Welcome to a discussion of the reticular formation, a system responsible for wakefulness, sleep, and coma, a system that extends from one end of the brainstem to the other and controls large portions of the brain's overall excitability. Our objectives are to identify the location of the reticular formation, describe the role of the reticular formation in consciousness, sleep, wakefulness, and coma, and to describe the common rhythms of the brain, alpha, beta, theta, and delta, of the normal electroencephalogram in terms of their frequency range, their distribution of the cortex, and their behavioral correlates. We'll look at the phasic events which occur in sleep during rapid eye movement, REM, and non-REM periods. We'll look at the neurotransmitters involved in the regulation of sleep and the function of the suprachiasmatic nucleus in the circadian rhythms. Why would we want to know about this? Because the reticular formation contains many of the structures that regulate consciousness, a critical feature of being alive. It is an important part of the system that regulates sleep and appetite. It serves as the source of many important neurotransmitters required by the rest of the brain. Understanding its anatomy and physiology will help us to understand why a patient is comatose, and many of the very common sleep disorders result from dysfunction of this region. Consciousness can be divided into two constructs: arousal, simply being awake, and content, having some substance to that wakefulness. Arousal without content defines the vegetative state, where somebody appears to be awake but has no content to relate to. Content does not appear to exist without arousal. Sleep is a special state in which the brain does not permit most of the motor output used to determine consciousness. Some definitions. Coma is a state in which no stimuli, whether internal or external, will arouse a patient to consciousness. Internal stimuli commonly include hunger and thirst, which activates a person normally, and external stimuli such as noise, light, verbal commands, and pain, which routinely will alert a person and bring them to consciousness. Stupor, the patient is unresponsive unless stimulated. Obtundation is when the patient is unresponsive but can arouse spontaneously. These patients may look as if they're asleep, but the EEG would show whether or not they're asleep. The EEG tells us about brain activity. The source of the EEG, or electroencephalogram, are EPSPs, excitatory postsynaptic potentials, and IPSPs, inhibitory postsynaptic potentials, over large numbers of neurons in the superficial cortical layers. The frequency ranges of these signals are the alpha wave, 8 to 13 hertz, beta waves, 14 to 30 hertz, theta waves, 4 to 7 hertz, delta waves, less than 4 hertz, and gamma waves, greater than 30 hertz. The normal EEG patterns include in a normal waking person, beta activity in the frontal lobes, and alpha activity in the occipital lobes. It's mostly symmetrical, and alpha waves block with eye opening due to input from the eye to the thalamus to the cortex. And there are commonly eye movement artifacts during wakefulness. During drowsiness, we'll see a loss of alpha and an increase in theta activity over the entire hemisphere, both hemispheres. During sleep, we'll see something called stage 2, in which there'll be distinct sleep spindles. As sleep gets deeper, we see slow wave sleep, in which delta waves predominate. And then during REM sleep, rapid eye movement sleep, it appears that the EEG is awake, and yet there are a lot of eye movements, and yet the person is otherwise asleep. We see here recorded from just one channel the electrical activity that might be seen at different levels of activation of the brain. During normal wakefulness, we might see low amplitude, high frequency activity, often called just desynchronized activity. During relaxation with eyes closed, we would see prominent alpha activity, particularly over the occipital lobes. As a person becomes drowsy, we'll see disappearance of alpha and the appearance of beta activity. And then a little bit deeper, starting to sleep, we'll see sleep spindles, as we see here, and some slower activity. And then in deeper sleep, we see that delta activity becomes more prominent. In coma, we may see unremitting delta activity. The normal EEG shown here is often recorded over a wide range of scalp locations, and we can see that during a wakefulness with eyes closed, we see a prominent alpha rhythm seen here over the left occipital lobe and the right occipital lobe. It's synchronized activity. And then when the eyes open, alpha is blocked because the visual pathway is activating visual cortex, and we see disappearance of alpha, desynchronized activity. As a person is asleep, we see slow wave sleep dominated by delta activity over the entire brain. The entire brain tends to go to sleep together. And then intermittently, we'll see rapid eye movement sleep in which the EEG becomes lower in amplitude, and there are obvious eye movements, artifacts detected near the front of the head. This non-REM, non-rapid eye movement sleep, and REM sleep are strikingly different in several regards. During non-REM sleep, also known as slow wave sleep, it's a deeper sleep, we see large, slow, synchronized activity dominated by delta activity. We see decreased muscle tone. We see it harder and harder to arouse a person. We may occasionally see dreams, but they're rather vague. And during that time, there is very much increased parasympathetic activity. And there's a slow, regular pulse and slow, regular respiration. During REM sleep, also known as paradoxical sleep, because it looks like the brain is awake from the EEG, there is low amplitude, high frequency, such as beta activity and desynchronized activity. The muscle tone is markedly abolished. It has the highest arousal threshold, that is, it's very difficult to awake a person at that point. There are often detailed visual emotional dreams, markedly increased sympathetic activity, and irregular pulse and respiration. There are variations in the sleep pattern throughout the night. If we look at the 8 hours of sleep, a person might start out awake with an awake EEG, and then over the next few minutes we'll gradually see a progression into deeper and deeper sleep. And then 90 minutes later, they'll cycle back up to come up to generate REM activity, that is rapid eye movement sleep with dreaming, and then cycle down the next 90 minutes down into slower and slower frequency where you see slow wave sleep, stage 4 sleep. And then another 90 minutes later, they'll cycle back up into a REM cycle with dream. And those cycles will continue throughout the night, the REM cycles getting longer and longer until the person finally wakes up. Somewhat similar cycling occurs throughout the day, but it's less dramatic. People don't necessarily sleep during the day, but their level of alertness may vary with a cycle of about 90 minutes. If we were to look over several days, as we see here, we can see that a person might be awake through good part of the day, and then gradually at night they cycle down through various levels of sleep. And then the next day they wake up, they're awake for long periods of time, and then they cycle down on a circadian rhythm. About every 24 hours they'll have a period of sleep and a period of wakefulness. There are many functions besides the obvious sleep and wakefulness that are linked to that cycle. That includes muscle tone, mental activity, autonomic activity, the pulse, respiration, temperature modulation, and hormonal release. You see just one of those, and that is the body temperature varying with a diurnal circadian rhythm, where it's highest while wakefulness and goes down in temperature during sleep, and it cycles back up and forth on a continuous basis following this sleep cycle that we defined in the EEG, along with muscle tone and other functions. That function that we've seen in a normal individual is destroyed by damage to the suprachiasmatic nucleus. Suprachiasmatic nucleus are in the hypothalamus, immediately above the chiasm, and destroying that destroys the normal sleep cycle. So that those functions that are linked, we now see that there's wakefulness and sleep that are intermixed throughout the day, and body temperature and all of those other functions that we identified are intermixed throughout the day, so you no longer have a diurnal cycle, a circadian cycle. So that circadian cycle is dependent on the suprachiasmatic nuclei, which get strong input from the visual system. Sleep disorders are a major source of disability, and they include difficulty initiating sleep, difficulty maintaining sleep, excessive daytime sleepiness, and sleep apnea, where there is problematic breathing at various stages of sleep. The reticular formation we want to discuss. The reticular is a resemble or forming a network, a meshwork. At most levels of the brainstem, it can be divided into three different longitudinal zones arranged in a medial to lateral sequence. The raphe nuclei, from the Greek word raphe, meaning seam, refer to the midline seam of the brainstem. They're thin plates of cells in and immediately adjacent to the sagittal plane, as we'll see. The medial zone, alongside the midline, are the raphe nuclei. They contain a mixture of large and small neuron, and it's the source of many long ascending and descending projections from the reticular formation. Some of the neurons in the medial zone of the reticular formation are so large that this area is referred to as the gigantocellular reticular nucleus. The lateral zone, which is particularly prominent in the rostral medulla and caudal zone, is primarily concerned with cranial nerve reflexes and visceral functions. We see here slices of different locations within the brainstem, starting from the caudal medulla, moving higher up in the medulla, to the pons and the midbrain. And we can see here in a lavender color in this tegmental region. This is the brainstem tegmental. We see a wide area of reticular formation. As we move a little bit higher up, we see it at the midsection of the medulla. It's seen here in the pons, just above the medial lemniscus. The medial lemniscus is seen here in blue, seen over in this portion of the pons, also just below the fourth ventricle. And then when we get up to the midbrain, we see that it is above the substantia nigra, and we see it in this portion of the midbrain as well, above the substantia nigra, medial to the medial lemniscus. The medial lemniscus is in blue, and the spinal thalamic tract in green. We, of course, see the peduncles here with the corticospinal tract in red, and the substantia nigra here in brown. We can see in these longitudinal view of the brainstem that the raphe nuclei are distributed just off the midline, from the medulla all the way up to the midbrain, to the upper pons midbrain. And we also see a number of reticular nuclei. They have various names here, and we're not interested in those. Just recognize that we'll refer to them as the reticular nuclei. And then if we look at cross-section through the brain, in this case the midbrain, we can see the raphe nuclei very close to the midline and the reticular nuclei more lateral. We see the raphe nuclei just below the cerebral aqueduct, adjacent to the periaqueductal gray. If we look at a longitudinal view for the brainstem, you can see some cells that are very large neurons, and they extend all the way from the pons down through the medulla and up through the midbrain, and they interact with several different places, and on the way down they will end up sending fibers all the way down to the spinal cord, to the anterior horn of the nuclei in the spinal cord, as well as up to the midbrain and the periaqueductal gray, to the hypothalamus and thalamus. We can see some of those pathways coming from the reticular formation, and we see the reticulospinal pathway coming down from the pons through the medulla all the way down to the anterior horn cells in the gray matter to influence the excitability of lower motor neurons. It turns out that the reticulospinal pathway is very important in controlling the excitability of those neurons, especially during sleep. And in fact, they inhibit the lower motor neurons through an interneuron in the gray matter of the spinal cord. We can see in this cross-section through the midbrain the location of some of the raphe nuclei. Don't worry about the details, just recognize that they're very close to the midline and close to the ventricle and the fourth ventricle, and just near the periaqueductal gray. But we can also see another nucleus of interest to us, and that is the locus ceruleus. And we'll be talking about that a little bit further in subsequent slides. It's also part of this system that's involved in controlling the excitability of the entire brain. We can see some of these nuclei, and we're not interested in the detailed name or location, but just recognize that we have here coming through the medulla, where we see the olive and the pyramids and the medial lemniscus right here. We see the reticular formation is in the tegmentum, and we see reticular formation in this location, in this location. As we come into the pons, we see locus ceruleus right near the periaqueductal gray, and again in the tegmentum, and the locus ceruleus will be of interest to us in influencing excitability of the brain, as we'll see shortly. In this mid-sagittal section, we can see that the locus ceruleus, located here in the pons near the midbrain, sends its axons to many different regions, including the thalamus, the hypothalamus, the amygdala, and the entire cerebrum, including the hippocampus, also the cerebellum, and that the reticular fibers descending the reticular spinal tract, traveling down to the spinal cord, will send long axons. The input from the locus ceruleus to the rest of the brain a good deal of it is carried through the medial forebrain bundle. We see here the distribution of some of the Raphe nuclei. Don't worry about the specific names of these different Raphe nuclei. Simply recognize that they're near the midline, in this case in the midbrain, right near the periaqueductal gray, just below the cerebral aqueduct, and we see them here in the pons, again right off near the midline. We see them in the lower pons, and then we see them in the medulla, again right at the midline. Here we see the projection of some of the Raphe nuclei, both to the spinal cord as well as to those vast regions of brain, cerebrum, as well as the septum, the thalamus, and the hypothalamus, to the amygdala, as well as to the hippocampus. Again, through the medial forebrain bundle, carrying much of the information to the forebrain. We see in this midline, mid-sagittal section, some important nuclei for us to recognize, and that is the Raphe nuclei, the locus ceruleus, the midbrain reticular formation, some hypothalamic nuclei, and the basal nucleus of Maynard. They're important because each one of them provides a distinctive neurotransmitter to wide portions of the nervous system involved in the overall activity of the brain rather than in a very selected pathway. In this case, we'll see that the Raphe nuclei utilize primarily serotonin. The locus ceruleus utilizes primarily norepinephrine. The lateral hypothalamus utilizes primarily orexin. The midbrain reticular formation and the basal nucleus of Maynard utilize primarily acetylcholine, and another nucleus in the hypothalamus uses primarily histamine. We're not interested in the details of these nuclei other than to know some of the neurotransmitters that they utilize, and that is orexin for the lateral hypothalamus and histamine for other sites within the hypothalamus. Here we see some of the interaction also within the preoptic area, and that is the preoptic area drives activity of many of these structures that we've just identified. In the preoptic area receives information that alters the excitability of all of those. That preoptic area that we've just referred to, the major neurotransmitter is GABA. And it turns out that these various neurotransmitters have markedly different activity at different levels of wakefulness and sleep. And notice that GABA is most prominent. GABA activation from the preoptic area is most active in sleep, whereas the other regions are most active in wakefulness. That is, all of these tend to be active. Orexin, histamine involved in hunger, histamine involved in activation, serotonin, norepinephrine, and acetylcholine, all increased in activity during wakefulness. Interestingly, the acetylcholine system involving the midbrain reticular formation is also very active in REM sleep. REM sleep being a condition that has some similarities to wakefulness in terms of the EEG itself. Now we see here the midbrain reticular formation projects both to the thalamus as well as to other portions of the brainstem. Unconsciousness. What is it? Consciousness itself depends on the interaction of the reticular formation and the cortex. There are only a small number of anatomical problems that can cause unconsciousness. Direct dysfunction of the reticular formation is one. Dysfunction of both cerebral hemispheres is the other. That is, damage to one hemisphere, if it does not affect the other hemisphere, does not produce unconsciousness. You can remove an entire hemisphere without altering consciousness. Whereas focal damage to the reticular formation can produce unconsciousness. And then if you disconnect these two systems, you can produce unconsciousness as well. How do we evaluate the comatose patients? The physical exam is focused on pupillary responses and extraocular movements. Why is that? Because these are largely dependent on structures within the brainstem. The extraocular movements that we look at are spontaneous roving movements of the eyes, cervical ocular reflexes if the cervical spinal cord spine is intact so that you can turn the head, the vestibular ocular reflexes, and vertical eye movements. These are measuring different portions of the brainstem from the third nerve all the way down to the sixth cranial nerve. Here we see one of the controls of pupil size, and that is the parasympathetic control of pupil size. That's dependent on light that activates the cranial nerves, 2, which activates Erdenscher-Westphal nucleus, portion of cranial nerve 3, and causes pupillary constriction. And you can see that damage in the midbrain would affect this pathway. Sympathetic control of the pupil comes from the hypothalamus through the lateral portion, as we'll see, descending sympathetic fibers in the lateral portion of the brainstem, which travel all the way down to the thoracic cord, and fibers travel out to the superior cervical sympathetic ganglion, where fibers travel up to the ciliary ganglion and then the short ciliary nerve to influence pupillary dilator, so that damage to the more lateral portion of the brainstem or any of this long pathway can control the size of the pupil under the sympathetic pathway. And then we can see that there are very many appearances of the pupils that give us some indication of where the damage is for influencing pupil size. If the damage is in the diencephalon, that is in the thalamus, you'll tend to see small pupils. If there is diffuse damage due to drugs, metabolic encephalopathy, the pupils will also be small and reactive. If there's damage in the pretectal area, in the midbrain, we tend to have large fixed pupils with hippus, oscillation. If there's damage in the pons, we'll tend to have pinpoint pupils. If there's damage in the midbrain, we'll tend to have mid-position fixed pupils. And if there's damage to the third nerve due to the herniation syndromes at the uncus, that is the temporal lobe pushing over the uncus into the midbrain, we'll tend to hit the third nerve and we'll have a dilated fixed pupil on that side. I don't want you to memorize these, but recognize that the pupils will give us some guide to location within the brainstem that might be explaining why somebody is unresponsive, that is unconscious. We see here some of the extensive structures involved in activation of eye movements to one side or another that involve activation of cranial nerve 8, turning of the head, that might stimulate cranial nerve 6, which sends fibers to the opposite cranial nerve 6, as well as to the third nerve through the medial longitudinal fasciculus, and that if there's damage to any portion of this pathway, there might be inability to move the eyes in the normal position when you turn the head. We see some aspects of that normal situation here, where the cervical ocular reflex is in a neutral position, that is the eyes looking straight ahead when the head is straight ahead. If you turn the head to one side, then you'll have the eyes tend to move in the opposite direction. If you move the head back, then you'll have the eyes tend to look down. Here we see somebody with an acute Wernicke's encephalopathy with small petechia hemorrhages in the pontine tegmentum just beneath the fourth ventricle. These affect some of those descending pathways between connecting the third and sixth cranial nerves through the medial longitudinal fasciculus, affecting eye movements and the coordination of those eye movements. The evaluation of the comatose patient can also be considered utilizing respiratory pattern to suggest location of damage that might be causing coma in a particular person. And that includes something called change Stokes respiration, central hyperventilation, apneustic respiration, ataxic respiration, and apnea, that is our lack of breathing. We see some of those patterns, and we just want to recognize that different kinds of patterns, you don't have to memorize these, but just different kinds of patterns suggest different locations that are causing coma in this individual. It could be in the thalamus, which of course normally gets input from the brainstem reticular formation, and in the thalamus you tend to have rapid breathing and then decreased breathing, rapid breathing and then decreased breathing. Damage to the a little bit lower in the midbrain, just below the periaqueductal gray in the reticular formation, tends to produce steady, rapid breathing. Damage a little bit lower in the pons tends to produce this slow, long, slow breathing. Damage further down in the pons tends to produce bursts of rapid breathing associated with apnea periods. And then damage in the medulla in the lateral portions bilaterally tends to eliminate respiration. We can also evaluate the comatose patient for motor responses. Normally, if you touch a person, there might be a defensive or localizing response suggesting that the corticospinal tract is functioning, which of course controls lower motor neurons. If there's a withdrawal, a reflex, then you know that the corticospinal tract controls some of those reflexes, and you have hyperreflexia if you had damage to the corticospinal tract. But in terms of specific location within the brainstem, we're particularly interested in whether we see decorticate posturing or decerebrate posturing. If we see decorticate posturing, that is where the arm is flexed, then it indicates that the damage to the motor pathway is above the rubrospinal tract origin. The rubrospinal tract origin is in the red nucleus in the midbrain, so that if the patient has a tonically flexed posture in the arm, it suggests that the damage is in the corticospinal tract above the midbrain. Whereas if there is extensor posturing, that is, the arm is extended, the legs are extended, then it indicates that the damage is below the red nucleus, that is below the midbrain, and that the vestibulospinal tract is dominating and is still active. The vestibulospinal tract is at the pontomedullary junction. It starts out in the vestibular nuclei near the pontomedullary junction. So if there's a decerebrate posturing, it indicates that there is damage just above the pontomedullary junction. And if it's bilateral, it's at the midline. Take a look at that. So here we see a situation in which you have perhaps an infarct or damage to one hemisphere, and that results in a flexor posture of the arm, which tells us that the damage is to the corticospinal tract, but that the red nucleus, which is in the midbrain, is still intact, and it's able to keep this arm flexed tonically. If there is bilateral damage to the corticospinal tract above the red nucleus, that is above the midbrain, then you will see bilateral flexion of the arms and extension of the legs. It tells us that the damage is just above the midbrain, leaving the red nucleus intact. If, on the other hand, the damage is lower in the pons, eliminating the red nucleus, but above the vestibular spinal tract origin of the vestibular nuclei, then we'll see bilateral extension of the arms and the legs, which tells us the damage is below the midbrain, entering into the pons, and leaving the medulla intact enough to produce those arm extension. But that means it's getting into an area close to the medulla, which is where respiratory centers are located, and putting those respiratory centers at risk. That's a dire situation. People generally die if that is not corrected. Damasio and colleagues examined a number of patients who were comatose to determine where their lesions were located. And in this setting, we can see the convergence of all of those different regions. We see the overlap in red of where most of the patients had damage, and we can see from the midbrain down through the pons that most of the damage was in this tegmental region, and particularly near the midline, but extended much more laterally than the midline, and it was right in what we would have referred to as the reticular formation. And we see it here in the midbrain, pons, in central regions. There are a number of historical views of coma. In Hippocratic times, people recognized that the brain was important for consciousness. Coma was viewed as a form of sleep until the 19th century. Several authors thought that coma with supratentorial lesions resulted from dysfunction of the cortex. And prior to CT scanning, almost all data was obtained from autopsies. And then gradually herniation came to be appreciated. Herniation occurs when pressure forces brain tissue to shift from one compartment in the brain to another, and can cause unconsciousness when pressure on the brainstem disrupts the reticular formation. And of course, the total volume of the cranial vault is limited, and that is the skull is not flexible, so that if something is growing inside the brain, whether it's a hemorrhage or a tumor, then the remaining tissue must shift position. Initially, a mass lesion displaces CSF, cerebrospinal fluid, but once the limit is reached and intracranial pressure increases, brain tissue, which is not elastic, causes herniation. That is, it shifts cone of tissue, forcing the brain out towards its only exit. The only exit is the foramen magnum, that is down from the cerebrum down towards the brainstem and out to the foramen magnum. Uncal herniation occurs when the medial temporal lobe, that is the uncus, was forced over the tentorial edge into the space beside the lateral midbrain. This can actually occur just before death. That is, when there's a mass in above the tentorium, then whether it's in the temporal lobe, frontal lobe, it can push the temporal lobe into the midbrain and produce brainstem damage, which can subsequently produce coma or eventually death. That herniation would compress the third cranial nerve, especially the parasympathetic fibers, to the pupil that travel on the outside of the nerve. This mechanism was thought to cause the unilateral dilation of the pupil on the side of the lesion, the so-called blown pupil, which is taken as an indicator of a herniation and of potential damage to the brainstem, starting out with the third nerve. The model suggested that there was a lateral pressure from the temporal lobe on the uncus, pressing on the brainstem, hitting between the thalamus and the midbrain and hitting the third nerve. And we see here the third nerve that might be pressed on by the uncus coming over the tentorial notch. We can see an example of herniation, in this case with a mass lesion in a supratentorial lesion that is distorting brain across the midline, and we can see that there's pressure right on the brainstem at this location, this being the pons. It's coming in and between the midbrain and the pons, as well as structures just above it. We can also see a hemorrhagic contusion in this setting, in which there's pressure, expansion of brain that pressed and distorted the entire brain to press over on the brainstem, pressing on the cerebral peduncle. And of course, the third nerve is coming out right between the peduncles. We can also see in this brain abscess a setting in which there's expansion of this portion of the brain, of this hemisphere, pushing all of the midline contents across the midline, but particularly distorting the brainstem at the level of at the midbrain pontine region. So you have an enlarged cistern. We should also recognize certain coma lookalikes. One in particular is the locked-in syndrome. In this situation, patients are awake, fully awake, but they cannot move their face, arms, or legs because they have damage to their corticospinal tract. In basilar artery thrombosis that leads to a pontine infarction, only the vertical eye movements may be spared because the basis pontis is eliminated, as well as control of facial muscles other than the eyes. You may also see other conditions that look somewhat similar, and that is in a severe Guillain-Barré syndrome, it may eliminate all movements because of involved in a more peripheral motor neurons. That includes pupillary reactivity. You may need EEG monitoring to see whether the patient is alive. And if that were Guillain-Barré, they might still be able to have a reactive EEG because it was more peripheral nerves and cranial nerves that were involved. The take home from all of this is that normal sleep-wake behavior is dependent on reticular formation and the suprachiasmatic nuclei in the hypothalamus. Consciousness is dependent on the reticular activating system, activating the cerebral cortex. Damage to these structures may result in sleep disorders or to coma. If you have any questions about this material, don't hesitate to give me a call.